Hello everyone. Today we will discuss about cell. First of all, my discuss will be in these parts. First, cell, plasma membrane, nucleus, two types of osmosis, cell organelles, mitochondria, plastid, endoplasmic reticulum, Golgi apparatus, lysosome, and lastly GERL system. And there will be two types of differences: plant cells versus animal cell and prokaryotic versus eukaryotic cell. First of all, I would like to draw your attention on cell. Cell is the fundamental structural and functional unit of living organism. The basic function of cell is to replicate spontaneously and to transmit hereditary information from parents to offspring. Cell has different types of cell organelles to perform different types of functions. Some cell organelles are responsible to digest food, and some cell organelles are responsible to respirate, to excrete, and to pump heart, to convey blood one part of the body to another. Now, the basic genetic material of cell is nucleus, and cell's outer covering is plasma membrane. Plasma membrane has a basic fluid mosaic model structure where lipid-protein lipid structure is seen here in this picture. The lipid is phospholipid and the outer section of lipid is hydrophilic, which is attracted to water. These two portions are exterior, so these are hydrophilic. The interior portion, this part is hydrophobic, not attracted to the water. In such a way, protein can be described as the same. The outer section protein is exterior protein, which is obviously hydrophilic, and the interior protein, this is interior protein and this is exterior protein. Interior protein is obviously hydrophobic. There is another type of protein which is transmembrane protein embedded like this with exterior part and interior part. This is transmembrane protein. Now, this plasma membrane is seen in plant cells and animal cells. Plasma membrane gives a specific shape to the cell and acts as mechanical barrier. Through plasma membrane, different types of materials are transported like food materials, nutrition specific materials, waste products, gases, oxygen, carbon dioxide, etc. Here, this transportation process is seen by diffusion and osmosis. Diffusion is the movement of substances from higher concentration to lower concentration. Suppose oxygen enters into the cell. Here, oxygen is in higher concentration outside the cell and is in lower concentration inside the cell. So, oxygen moves from high concentration to low concentration into the cell. And carbon dioxide, on the other hand, exits the cell through cell membrane. So, carbon dioxide we can say is in high concentration inside the cell. Due to the different types of reactions, carbon dioxide is produced. So, it is higher concentration inside the cell and in low concentration outside the cell. That's why carbon dioxide moves from high concentration to low concentration and moves out the cell membrane. Now, I would like to draw your attention on osmosis. There are two types of osmosis, exoosmosis and endoosmosis. Actually, osmosis is the movement of water from high concentration to low concentration. When the cell is kept in the hypotonic solution, means where water concentration is high. This is a hypotonic solution and water concentration is high outside the cell. So water enters into the cell and the process is endoosmosis because water moves in the cell. So this is endoosmosis like this way. So the cell actually engulfing water bursts out, and on the other hand, if cell is placed in the hypertonic solution, and this is hypertonic solution where the water is in less concentration, okay, water is high concentration inside the cell, so we can say water expels out the cell membrane, so exoosmosis occurs, and that's why after exoosmosis cell shrinks. So in such a way, the osmosis diffusion works on the plasma membrane to transport different types of materials. Especially in plant cells, cell wall can be seen. Cell wall is a rigid, flexible membrane outside the plasma membrane, and there is deposition of cellulose, hemicellulose and pectin or sometimes lignin, suberin, whatever. Actually the main function of cell wall is to give rigidity, flexibility and mechanical support to the cell. Now we can say about nucleus. Nucleus is the genetic material double layered cell organelle where nuclear membrane is double layered, and inside this nucleus there is a specific cytoplasm called nucleoplasm, and chromatin fiber is present, and there is a dense round shaped structure called nucleolus. Here it is the nuclear membrane which is porous, and inside nucleus there is nucleoplasm, and this is round shaped dense nucleolus, and these are chromatin fibers which is condensed to form chromosome during cell division. The main function of nucleus is to replicate spontaneously during cell division and to transmit hereditary information from parents to offspring. In prokaryotic cell, the DNA is not combined with the histone protein. So DNA, here it is naked gene or nucleoid. But in eukaryotic cell, we can see the solenoid model structure. DNA is combined with histone protein. So this type of nucleus is found in the eukaryotic cell. In prokaryotic cell, there is no nuclear membrane, but in eukaryotic cell, we can see the nuclear membrane covering the nucleus. Now, cytoplasm. Cytoplasm actually has different types of cell organelles, and the basic powerhouse of the cell organelle is mitochondria. And mitochondria is covered by double layer membrane, and this is outer membrane and this is inner membrane, and between these two membranes there is intermembranous space, and from inner membrane there is a finger-like projection called cristae, and the inner matrix is called stroma. So this is the basic structure of mitochondria, and actually through respiration mitochondria can generate energy in the form of adenosine triphosphate ATP, and we can use this energy for our daily work. The another cell organelle is the kitchen of the cell called plastid, and this plastid is also covered by a double layer membrane. Internal matrix is called stroma, and inside the stroma there is granum stack forming grana. These are granum stack and forming grana. Here green colored pigment chlorophyll is found, which is responsible for the photosynthesis of the plant cell, and this type of plastid is called chloroplastid. when we can find as pigment there is carotenoid or xanthophyll, the plastid is chromoplastid. On the other hand, if this plastid is responsible to store starch, then this is amidoplast. When the plastid is responsible to store protein, this is elaioplast, and aleuronplast can store lipid in it. This amidoplast, elaioplast and aleuronplast are leucoplast because they can store food materials, nutritive materials in it and white in color. So this is the basic structure and functional characteristics of plastid, which is kitchen of the cell. And lastly, we can see, I would like to draw your attention on these three types of cell organelles, which are endoplasmic reticulum, Golgi apparatus and lysosome. Lastly, I told you that here it is the nucleolus. This nucleolus actually responsible for the synthesis of ribosome. From nucleus, this ribosome is attached to the endoplasmic reticulum. Endoplasmic reticulum is a tubular-like or branched-like or vesicular structure. This is tube, this is branched, and this is vesicle structure. And if ribosome is attached to this endoplasmic reticulum, then ER is called RER, rough endoplasmic reticulum. This rough endoplasmic reticulum is responsible for the synthesis of protein. When there is lack of ribosome outside the membrane of endoplasmic reticulum, then this will be called smooth endoplasmic reticulum, which is responsible for the synthesis of fat. And SER also can detoxify the drug and heavy metals in the liver. So, this is the main function of RER and ACR. Now, from RER, the protein is polypeptide chain is synthesized and is packed in the vesicle, and this vesicle convey this polypeptide chain to the Golgi apparatus. Golgi apparatus has a basic structure. This is a small vesicle, this is large fluid-filled vacuole, and this is cisternae. Cisternae has C phase and trans phase, and endoplasmic reticulum, this vesicle convey the polypeptide chain to the cis phase, and within this cisternae the polypeptide chain modification occurs, and from trans phase it is transported to lysosome, and lysosome is the housekeeper of the cell. Lysosome is the suicidal bag in which the residual body waste part is engulfed by the cell organelle. Now, protein synthesis in the form of polypeptide chain, protein synthesis with modification, all this process happens to Golgi apparatus, endoplasmic reticulum and lysosome. GERL system is formed. Now we can summarize the whole discussion in cell, plasma membrane, nucleus, cytoplasm, mitochondria, plastid, Golgi apparatus, lysosome, and forming GERL system. With the difference of prokaryotic cell and eukaryotic cell, we can say that there will be lots of questions around 5 marks, 3 marks or 2 marks. I think this video, these informations, this discussion will be useful to you, and if you like this video, then please download Google Play Store Knowledge Flame app, and please subscribe YouTube Knowledge Flame channel.